Good afternoon, dear students. Lecture number 3, the topic of the lecture, Gene Level of Organization of Hereditary Material, Model Theory of a Gene, Implementation of Genetic Information into a Sign. Plan of the lecture. At first, gene definition at different stages of biology development, modern definition of a gene. And the next one, properties and classification of a genes. Genetic code properties. The next one, genome, genotype, karyotype, International Human Genome Project, human genome sequencing and mapping. Stage of genetic information realization, protein biosynthesis. In conclusion, central dogma of molecular biology. There are 3 levels in genetic material in the cell: genetic, chromosomal, genomic. At the last lecture, we found out the molecular basis of heredity. Today, we will study a genetic level. In 1866, Gregor Mendel suggested that characters of organisms are determined by inherited units, which he called hereditary traits. Afterwards, they were named genes. It is very difficult to give the exact definition of a gene, but let's try to do it. The first version is a Thomas Morgan version. He told about a gene as a unit of recombination. A gene is the smallest region of a chromosome that can be separated from adjacent regions as a result of the cross-linking. But in this case, a gene is a big fragment of a chromosome. The next definition is gene is a mutating unit. A gene is the smallest region of a chromosome capable of a mutation. But in this case, only one pair of nucleotide can be considered a gene. You know, there are the point mutations, so this definition is not full and it is not quite right. The third definition is a Biddle and Tatum version, which explored a gene as a unit of function. It is the smallest region of a chromosome, that it is causing synthesis of a certain product. It may be a protein or enzyme. This is the Biddle and Tatum concept: one gene, one enzyme. Afterwards, it was converted in concept one gene, one polypeptide, and later one cistron, one polypeptide. The cistron is a fragment of a DNA molecule which includes the necessary information of one polypeptide strand synthesis. Well, the summary and the modern Human Genome Project definition of a gene. A gene is a DNA transcription site that is translated into one or more amino acid sequences of a polypeptide or nucleotide sequences of a tRNA and a rRNA. The properties of a gene are discretization or discreteness. A gene is the molecular base of a distinctive feature of the organism's phenotype. Secondly, specificity. A gene is the information carrier of amino acid sequence of a specific polypeptide. And the next one, pleiotropy. The multitypal action. A gene takes a part in to forming of many specific features of organisms. Dosage of action. This is a quantitative dependence of the results of gene expression on the number of dominant alleles of the gene in the genotype. So, at the genetic level of hereditary material organization are provided individual inheritance and individual changes in characteristics and properties of cells and organisms of these species. Classification of the genes. The first group is RNA-coding genes, encode the synthesis of tRNA, rRNA and mRNA. And the second group is protein-coding genes. Household genes encode proteins that support cell life. Terminal differentiation genes encode proteins responsible for cell and tissue differentiation, respectively performing specific functions. And transcriptional factors genes encode proteins involved in the transcription process. And there are also the mitochondrial genes, code of oxidative phosphorylation proteins, tRNA and rRNA. And the next point of the lecture is genetic code. I think all of you know about the genetic code. Remind you that biological information in nucleic code system presented by the nucleotide sequence and in protein code system by the amino acid sequence. So the protein structure basis is on an amino acid sequence which based on DNA nucleotide sequence with the help of genetic code. There are 4 nucleotide types: adenine, guanine, thymine, and cytosine, forming the 64 triplets which code all 20 amino acids. Each amino acid is coded by 3 nucleotides. Properties of the genetic code: triplet structure. A code is a triplet of nitrogen bases of DNA, polynucleotide chain. Universality. The genetic code is universal for all organisms, the same 4 nucleotides and the same amino acids. Degeneracy or redundant. The same amino acid can code more than 1 triplet. Specificity. Each other triplet code only 1 amino acid and non-overlapping. A code doesn't cross. Each nitrogen base belongs to only 1 triplet. Okay, we studied and repeated the genetic level of genetic material organization. Let's talk about chromosomal level. The genetic material of eukaryotes in interphase nucleus is presented by chromatin. When cell divides by mitosis, the chromatin is spiralized to chromosomes. Besides DNA, chromatin contains many different proteins. Most of them are histones. They have 5 classes: H1, H2A, H2B, H3, and H4. There are 4 levels of chromatin folding. The nucleosome string. This level of chromatin organization is provided by 4 types of histone proteins: H2A, H2B, H3, and H4. They form a protein's body that look like a pack, the cores. The cores are formed from 8 histones, 2 of each type. The DNA molecule spirally turns out the protein's core. The cores are connected with each other by linked DNA. The linked DNA may be 15 to 100 nucleotides long. It depends on cell type. Chromatin fibril. Next chromatin folding is provided by H1 histone protein. It is bonded with linked DNA and it puts nucleosomes close to each other. Such chromatin fibril, so-called elementary fibril, has a diameter 20 to 30 nanometers and length 1 to 1.2 millimeters. Interphase chromonym. This level of chromatin folding is provided by folding of chromatin fibrils to loops. The non-histone proteins take a part on this process. They merge pointed regions, making the loop with the fragments of chromatin fibril in it. The one loop contains from 20,000 to 80,000 nucleotide pairs. After such folding, interphase chromonym has the diameter 100 to 200 nanometers. The regions of interphase chromonym undergoing further folding makes a structural blocks, which can be visible in the interphase nucleus as chromatin particles. There are euchromatin regions and heterochromatin regions, according to their functional activity. The euchromatin regions have a less tight folding because of active transcription process. The heterochromatin regions have a tighter folding because of lack of the transcription processes. There is a constitutive and facultative heterochromatin. The constitutive heterochromatin is in the telomere regions and regions near the centromere and along some internal fragments. It is believed constitutive heterochromatin to provide keeping of total nucleus, shape attaching chromatin to carriolem, participating in chromosome recognition during meiosis, making in intervals between genes. The facultative heterochromatin has information. It contains genes and may be changed to euchromatin. The facultative chromatin formation occurs during process of cell differentiation, serves as a mechanism of switching off activity of several genes, which is not necessary in the cell of such spiralization metaphase chromosome. When the cell is preparing to divide, we can't see the chromosomes in microscope very well. DNA molecule connect with histones and it is not transcribed and it is not duplicated. In the beginning of mitosis, chromatin condenses to chromosomes. During the protein biosynthesis process, linkage between DNA and histone destroys. At the same time, chromatin structures and the chromosomes become visible in a microscope. It is a distinguish between genome, genotype, and karyotype. A genome is a collection of genes in the haploid set of chromosomes of cells of a given species. The physical size of the genome is 3.3 billion pairs of nucleotides. A genome is a collection of genes included in the chromosomal set. A combination of unique genomes derived from each parent creates the genotype underlying genetic individuality. A karyotype is a collection of morphological features of a complete chromosomal set characteristics of cells of one organism of a given species. The progress of Human Genome Project is completely sequenced genome, deciphered sequence of human DNA nucleotides, and gene mapping, location of certain genes of certain sites of chromosomes has been determined. The Human Genome Project developed in 1966 in the United States on the initiative of James Watson. The program manager was Francis Collins, American Genetics, and the first person whose genome was completely sequenced was James Watson. This is a very time-consuming and expensive procedure. The first sequencing cost $3 billion, and at the end of the program, the cost of the sequencing a single genome was about $100 million. As of 2015, the cost of a complete genome reading was about $5,000, and the cost of analyzing only selected section of the genome now about hundreds of dollars already. Currently, the trend is the speed of sequencing increases, but price fall. There is a genetic passport in several countries: USA, Canada, European countries. It includes information about human genome mutation, predisposing to hereditary and multifactorial diseases. This is the base of preventive and individualized medicine. And the next point of the lecture is the protein biosynthesis. The stages of biosynthesis of protein are transcription, translation. Transcription is the mechanism by which a single-strand DNA-based sequence is rewriting to a complementary pro-mRNA-based sequence. In this fragment of DNA, histones separate and the strands of DNA open to the polynucleotide sequences. Hydrogen bonds between nitrogen bases get destroyed with the help of helicase enzyme, and the separate chains forms. One of these chains becomes the matrix for the building pre-matrix RNA or pre-messenger RNA. The pre-mRNA molecule forms as a result of connection each other three nucleotides by the RNA polymerase by the rules of complementarity. Well, let's watch the video. The pre-mRNA gets processing and splicing. In the course of processing, introns exist by the restriction enzymes. Adjacent exons are cross-linked by the enzymes of ligases. So mRNA or messenger RNA is formed. The next phase of the protein biosynthesis is translation. Translation is the process when the base triplet sequence in mRNA molecules is translated into a specific amino acid sequence in the polypeptide chain. This process is on the ribosome or polysomes. Each ribosome consists of big and small subunit. mRNA connects with the small subunit of ribosome until its first codons, triplets, turn to the big subunit. The first codon bonds a tRNA molecule, which includes its complementarity anticodon and holds the first amino acid, usually methionine. After it, the second codon connects with next amino acid tRNA complex, which includes anticodon complementary of this codon. The function of ribosome is hold on necessary position to mRNA and translation proteins factors until the linkage between amino acid by the peptide bond. When the new amino acid connects with the growing peptide chain, the ribosome moves on the mRNA to the new codon in the right place. When the tRNA becomes free from the amino acid, it leaves the ribosome and comes back to the cytoplasm to form the complex with the new amino acid. The reading of mRNA text by the ribosome continues until one of the stop codon, triplets, or uracil adenine, uracil adenine guanine, and uracil guanine adenine. On this stage, the polypeptide chain leaves the ribosome. So, the translation stages are binding of the mRNA to the ribosome, activation of the amino acid and its binding to the tRNA, initiation, start of the polypeptide chain synthesis, elongation of the chain, termination of the chain synthesis, further use or destruction of mRNA. After separating peptide chains from ribosomes, they acquire a secondary, tertiary, quaternary structure. This is a post-translational modification of proteins. So, the protein biosynthesis is under the direct control of the genetic material of the cell. Okay, let's watch the video and try to remember the cell structures which take a part in transmission of hereditary information. Try to determine the stages of the protein biosynthesis.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
The central dogma of molecular biology was formulated by Francis Crick. All genetic information encoded in the primary structure of DNA is realized through the process of protein biosynthesis. The formed proteins performing various functions are regulators of metabolic process, the combination of which ensure the formation of a certain trait. Thank you for your attention.